Good afternoon. My name is Tairine, and today I will present the article entitled Genomic and Transcriptomic Basis of Hanseniaspora vineae, Impact on Flavor Diversity and Wine Quality, accomplished by Giorello and collaborators. In the abstract, we did not find the objective of the article clearly, but it is understood that they are looking for a new species, Hanseniaspora vineae, to produce quality wine. Here we see the basic design of the article in abstract. Major finds is in here, and here we have the interpretation and conclusions of the authors. According to the CARS model, the introduction of this article includes Move 1, Move 1 mixed with Move 2. Move 2 is not a clearly, it's mixed with a hypothesis of this work. Move 1 again and ends with Move 3.
Yeast are responsible for transforming a simple grape must into wine. Alcoholic fermentation, the process of converting sugar into alcohol and the carbon dioxide, it's only possible thanks to yeasts. Its role in the world of wine, however, is not restricted to this fact alone, but also to a wide range of influences on flavor and aromas that are being increasingly investigated and detailed by winemakers and researchers. In addition to the convert sugar into alcohol, yeast also produce products such as ester, aldehydes, monoterpenes, among others. This is just about sugar metabolism. However, yeast are still able to interact with nitrogen, sulfur, and other phenolic compounds, in addition to metabolize compounds related to flavor and create others during their autolysis. All of this needs to be taken into account by the winemaker when the wine is being produced. Therefore, the choice of yeast to be used is the crucial point.
In wine, non-Saccharomyces yeast strains that account for more than 99% of the grape native flora are still poorly explored, and their impact on flavor richness will require multidisciplinary studies, from genetics to metabolic analysis of the yeast cells. The limited number of commercial yeast strains applied by today's winemakers are not contribution to flavor diversity, decreasing the possibilities to obtain more differentiated wine styles. Besides the grape selection and viticulture and vinification technologies used, which have been traditionally emphasized for quality wine production, this aspect should be considered. In a highly competitive market with more than 1 million brands of wine, differentiation and increased flavor diversity will be obtained with the application of increased yeast diversity during the fermentation process. Non-Saccharomyces species of yeast have been reported as beneficial for winemaking because they contribute to the sensory complexity of wines. Recent Hanseniaspora vineae demonstrate the ability to provide increased levels of acetato esters and benzoids and decreased levels of high alcohols in wine by pure fermentation or by co-fermentation with Saccharomyces cerevisiae. Higher alcohol formation via the Ehrlich pathway is subdivided in 3 steps: transamination, decarboxylation, and reduction, like seen in the figure. In transamination, the key enzymes are the branched chain amino acid transaminases and the aromatic amino acid aminotransferases, which catalyze the transfer of amines between amino acids and their respective alpha-keto acid. In the second step, the alpha-keto acids are decarboxylated through pyruvato decarboxylases to form their respective aldehydes. Finally, the reduction from aldehydes to alcohol is carried out by alcohol dehydrogenases, and aryl alcohol dehydrogenases. The formation of the fruit and flowery-like aroma acetato esters is dependent on acetato and alcohols, and they are due to 2-alcohol acetyltransferases encoded by 8F genes in Saccharomyces cerevisiae. The biosynthesis of ester is carried out by 2-acyl coenzyme A or ethanol O acetyltransferases and involves ethanol and acyl coenzyme A units. Ethyl ester as well as acetate esters contribute fruity-like aromas. And day concentration levels in wine are significantly lower than those of the acetate esters. Thus, the main goal of this article is to understand the alternative metabolic pathway of Hanseniaspora vinea compared to Saccharomyces cerevisiae pathway, which will contribute to a better understanding of biology of apiculate yeast, the main group of yeast associated with fruit. In material and methods, we have the biological material. The authors compared two yeast strains of Hanseniaspora vinea with 14 commercial Saccharomyces cerevisiae. To understand which group of yeast the strains belong, firstly the DNA and RNA isolation of Hanseniaspora vinea strains was carried out for vineyards in Uruguay. These strains were identified by sequencing the ribosomal region and amplification by PCR technique. The author stimulated the genome length employed by mass cytometry. The results were subjected to analysis with appropriate software. All analyses were performed in triplicates. With the genome sequenced, the authors created a genomic library with the genes found. With the help of specific software, they also mapped short genomic sequences to estimate the diversity between the two strands of Hanseniaspora vinea. For several genes with no function in the biosynthesis of acetato, ethyl ester, and higher alcohols, they determined which ones were present, duplicated, or absent for the Hanseniaspora vinea genome compared to the commercial strains of Saccharomyces cerevisiae. The authors, using a public database, analyzed clusters of orthologists using proteins as a base. For the phylogenetic analysis, maximum likelihood was performed, concatenated with a set with 227 genes for 29 species. The transcriptome were analyzed in 3 fermentation stages, with 1, 4, and 10 days. The readings were sequenced and analyzed. The respective counts obtained for each gene were then used to calculate the differential expression of each gene using specific software. They yielded a total of 7.8 gigabytes of data and 52 million paired reads. The transcriptomic reference construct resulted in 4,725 contigs with an average and median length of 1,982 and 1,683 pairs base respective. The authors carried out tests of aromatic compounds in a synthetic medium under chemical fermentation conditions, simulating vineae. Samples were taken 4 and 10 days during fermentation and in the final fermentation process. The volatile aroma compounds were extracted and analyzed using a mass spectrometer. Thus, these components were identified and quantified, compared with the pure wine aromas also stored in databases. Alchemical analyses were performed in triplicates. Analysis of variance was performed to determine the difference in the concentrations of aroma components between strains using statistical software. The sequences obtained from the complete genome were deposited in the NCBI database, GenBank. Results and discussion. The genome analysis revealed high similarity in both genomes of strains of Hanseniaspora vineae about size and the prediction of genes. With the comparison made with the commercial strains of Saccharomyces cerevisiae, the authors found 3,861 homolog sequences of the genes and more than 4,141 sequences aligned to the NCBI of proteins. The genes related to sterol compound synthesis in Hanseniaspora vineae were compared with those reported for Saccharomyces cerevisiae. Absent homologies and repeated genes were found. Example: in Hanseniaspora vineae, we have 3 copies of ARO8 gene. However, BAT2 gene is absent in comparison with Saccharomyces cerevisiae. Several genetic and phenomic characterizations were analyzed to compare Hanseniaspora vineae and Saccharomyces cerevisiae strains. The aroma components profiles determined by gas chromatographic mass spectrometry of Hanseniaspora vineae were compared with those of Saccharomyces cerevisiae and are shown here in Table 5. For example, only was identified in Hanseniaspora vineae compounds like 2-methyl-2-butanol, 2,3-butanediol, benzyl alcohol, and benzyl acetate. On the other hand, in Table 4, with the same aroma compounds produced by Hanseniaspora vineae at days 4 and 10 are shown, and the differential profile of genes involved in high alcohol, acetato ester, and ethyl ester metabolism were evaluated. For example, 2-ethyl-1-hexanol, ethyl lactate, ethyl-2-hydroxyhexanoate, benzyl acetate, ethyl-4-hydroxybutanoate, diethyl-2-hydroxyglutarate is detected only in day 10. The authors, through comparison with other genes, found that sequenced genome was 98% complete, and that Hanseniaspora vineae and Saccharomyces cerevisiae use only 52 similar, showing a grand diversion between species. In addition, heterozygosity can be evidenced by the analysis of the single nucleotide polymorphism using the different strains of Saccharomyces cerevisiae. Between the two strains of Hanseniaspora vineae, the nucleotide diversity was 1 variant per 179 bases pair. By concatenation, the 127 genes of 29 species, they classified Hanseniaspora vineae as part of Saccharomyces complex, but out of the whole genome duplication clade, showing that Hanseniaspora vineae previously diverged for the whole genome duplication clade and emergent for the CTG fungal clade. In this image, we have in pink the CTG clade, fungal clade, in green Saccharomyces complex, and the yellow whole genome duplication clade. And here in red we have the position of Hanseniaspora vineae inside the clade, this Saccharomyces complex. Using the chemical fermentation technique, the authors report similar amounts of genes that were expressed at the 3 fermentation points: exponential growth day 1, end of the exponential phase day 4, and end of the stationary phase day 10. More than 2,500 genes differently expressed With the assembly of the transcriptome, it was possible to identify 15 more genes than those obtained by genome analysis, and almost all those obtained were confirmed. Transcriptome analysis for days 1, 4, and 10 showed that 4,468 genes were common for the 3 periods. Important changes in gene expression were detected between any pair of the 3 fermentation points, while differences between repetitions were minimal. Hanseniaspora vineae compared with Saccharomyces cerevisiae, there was a considerably high expression on days 1 and 4 of genes related to biochemical growth cascades and glycolysis. In the figure C, we see which compounds are begin expressed and indicate period. In figure A, I see in A a diagram showing the differential expressed genes shared between each fermentation point, in B showing this gene shared between each fermentation point for the top 100 most highly expressed gene, and C, in figure C, main biochemical cascade of the most expressed genes of each sample day on fermentation. In day 1, have biosynthesis of amino acids, glycolysis, gluconeogenesis, methane metabolism, and pentose phosphate pathway being expressed. In day 4, steroid biosynthesis, arginine biosynthesis being expressed. Between day 1 and day 4 have pyruvato metabolism, synthesis of secondary metabolism, oxidative phosphorylation, citrate cycle (TCA), and glycine serine threonine metabolism being expressed. And day 10, protein processing in endoplasmic reticulum and GPA antibiosynthesis being expressed. The analysis of aroma compound showed that, in general, alcohol production was twice as high in Saccharomyces cerevisiae than in Hanseniaspora vineae, what may be happening due to the fact that the most important gene involved in the transfer that leads to the production of a higher alcohol in 2-phenylethanol is BAT2 in Saccharomyces cerevisiae. However, this gene is absent in Hanseniaspora vineae. In figure A, we have the comparison of total higher alcohols, 2-phenylethanol and phenylethyl acetate produced in Hanseniaspora vineae and Saccharomyces cerevisiae at day 10 of fermentation. In figure B, 3 steps of metabolic pathway of higher alcohol biosynthesis with putative enzymes involved in Hanseniaspora vineae. Here we see the absence of the gene BAT2. In figure C, production of total higher alcohols and 2-phenylacetate by Hanseniaspora vineae at 1, 4, and 10 days of fermentation. And in G, we have expression heat map of genes putatively involved in the higher alcohols and 2-phenylethanol production for Hanseniaspora vineae at 1, 4, and 10 days of fermentation. Higher correlates indicate higher expression values, and data are showing for duplicates. Significant changes in expression of a gene are indicated with arrows. Unlike what happens with the amount of 2-phenylethyl acetate, which is more produced by Hanseniaspora vineae, which have 3 copies of the ARO gene, while Saccharomyces cerevisiae has only 1 copy. In figure A, we have the cooperation of total acetate esters and 2-phenylethyl acetate produced in Hanseniaspora vineae, and Saccharomyces cerevisiae at day 10 of fermentation. We can see the production of 2-phenylethylacetate for Hanseniaspora vinho is higher. In figure B, we have the metabolic pathway of acetate ester biosynthesis with putative enzymes involved in Hanseniaspora vinho. We see the 3 copies of the gene. In figure C, we have production of total acetate ester and 2-phenylethylacetate by Hanseniaspora vinho at 1, 4, and 10 days of fermentation, and figure 3, expression heat map of genes putative involved in total acetate ester and 2-phenylethylacetate production for Hanseniaspora vinho at 1, 4, and 10 days of fermentation. Higher colors indicate higher expression values, and that are showing for 2 cases. Significant changes in expression of each gene are indicated with arrows. This also occurs with decarboxylation result in an increase in the intensity of the cyclorons. The absence of one of the main genes involved in ethyl ester production, EEB1, is in Hanseniaspora vinho is consistent with the results. Like we see in figure A, we have the comparison of ethyl ester production in Hanseniaspora vinho and Saccharomyces cerevisiae at day 10 of fermentation. In B, we have metabolic pathway of acetate ester biosynthesis with putative enzymes involved in Hanseniaspora vinho. You can see again the absence of the gene EEB1, this responsible for ethyl ester, one of the responsible. In figure C, we have the production of ethyl ester by Hanseniaspora vinho at 1, 4, and 10 days of fermentation. And in figure D, we have the expression heat map of genes putative involved in ethyl ester production for Hanseniaspora vinho at 1, 4, and 10 days of fermentation. High colors indicate high expression values, and dates showing are of duplicates. Significant changes in expression of each gene are indicated with arrows. To conclude, the use of non-Saccharomyces yeast in making is limited to the insufficient characterization of many species that naturally participate in the process. Hanseniaspora vinho provide to contribute to the diversity of flavors under winemaking conditions. The higher production of acetate esters, the increased ratio to phenylethyl acetate, and the reduced amount of 2-phenylethanol and ethyl ester found in Hanseniaspora vinho may be due to high presence of putative alcohol, acetyltransferases proteins, and absence of the EEB1 gene. Hanseniaspora vinho produce a large amount of phenylpropanoids compared to Saccharomyces cerevisiae and other yeast, which can be explained by duplication of genes and by highly expressed ARO genes. The authors also established with their work that Hanseniaspora vinho can be a potential model of eukaryotic species to study the benzoid synthesis pathway, phenolic compounds that have several functions in plants, which make them highly attractive to the yeast biotechnological industry.
This is all. Thank you all for watching.